Hi, everybody. My name is Sayali, and I'm a PhD candidate. I work with Dr. Daifeng Wang at the University of Wisconsin-Madison. And let me just, my talk today is based on this model, ARTEMIS, that I developed during my PhD. And a shortened version of the title that she just mentioned is modeling cell trajectories, population changes, and perturbation effects in time series single cell transcriptomics with ARTEMIS. And let me start today with some background on the data and the challenges that I addressed in this work. As we all know, with high throughput sequencing, we can profile thousands of cells to study complex biology of processes like developmental trajectories in embryogenesis or some organ development, or even to study disease progressions like cancer or Alzheimer's. But we also know that the sequencing is very destructive, which makes it impossible to accurately capture continuous measurements of cells, how cells actually change or evolve across time. So the key challenge that this introduces, because of this nature of sequencing, is the missing continuous measurements and alignment of cells across time points, right? So that leads to the question of how can we reconstruct this continuous trajectory underlying biological processes. And today I will focus mostly on the gene expression of these cells. And what we see here is just this snapshots of observed cells at these discrete time points. And for a given cell, each dot is a cell. So for a given cell, we don't know how this evolves over time in this biological process. And to address this, I developed ARTEMIS. So let's go through the problem setting in more detail. What we observe is gene expression at discrete time points. But what you see is that the cells at time t minus 1 are not exactly the same as those as time t. So there's no direct alignment. So the challenge here is to infer this unknown alignment to reconstruct this single cell trajectory. And also we have only static snapshots of cells. So even though they're different cells, we assume that they follow the same um, underlying evolutionary process. And meanwhile, the cell population itself is changing through birth, death, and proliferation and differentiation. And existing models that were developed to, again, model the cell trajectories don't really accommodate such population changes. And on top of that, we have very little or to no understanding of the actual dynamics that are driving these molecular processes or molecular changes, and there's no mechanistic method or model that we can just incorporate to guide us in this modeling. And finally, the cells that we start with might be like a homogeneous group, like some stem cells that over time they diversify into these different or distinct cell types, which further adds to the complexity of modeling the processes. And with that setup, the assumptions that we make or we can make in developing our model is that first, we know gene expression is high dimensional. So our data is high dimensional data captured at discrete time points. Second, we can assume that there's some underlying biological factor driving this progression of cells over time. Third, we assume that these cells change gradually and not abruptly. So we can infer some alignment that's also linear in nature. And we also assume that this evolution is like a stochastic process, which is coherent with the assumption, with the biology that we all know that cellular processes are inherently random. And finally, we assume that the number of cells or the sequence cells at each time point actually reflects the true relative population with minimal bias from any sequencing experimental noise. And based on this method, based on these assumptions, our method on a very high level looks like this, where we have these cell by gene matrices from observed time points, and this goes into the model, and the model tries to predict or recover the unmeasured cell by gene matrices in this process so that we can reconstruct the cellular trajectory. And more specifically, the model has two components: a variational autoencoder that is trained on the observed cell by gene matrices from the observed time points. And then we have the Schrödinger bridge, which learns like a stochastic process, and it uses this coupled system where we have two SDEs, forward and backward, that kind of that are optimized together iteratively and learn this trajectory in this latent space. So before I go into more details, let me just speak a few words about Schrödinger bridge. And so it's actually a concept that popped up from particle physics by Schrödinger, who is this famous physicist of the wave equation. And in Schrödinger bridges, what we want to learn is this stochastic process P over a time window from 0 to 1, such that this process starts from a distribution mu naught and ends at another distribution mu1 at time t. And to do this, we assume that this process is similar to some reference process q. And often this q can just be like some Brownian motion, which is like a prior of how particles might move. So given the starting distribution mu naught, a target distribution mu1, and some reference process q, we want to find this most likely stochastic path P, which connects these two distributions. And we can solve this, we can find this P by actually solving the scale divergence problem, which results in a formulation that closely resembles also another very famous concept, which is the entropy regularized optimal transport. But I'm skipping the math here. But however, solving this scale divergence problem is very complex. And thanks to advances in math and computer science, we can reduce it to a much simpler version that's easier to understand and implement. So the Schrödinger bridge, the solution to that scale divergence can be expressed through these coupled stochastic differential equations. There's a forward equation and a backward equation, and each equation is a process, and it's guided by some policy. And this system is called diffusion Schrödinger bridges, which was recently developed for again modeling cellular perturbations. But we are using this for this reconstructing cellular trajectories. But what these equations mean is that, for example, the forward SDE means that if I start from time 0 with mu 0 or mu naught, and I follow this forward policy, I evolve to mu 1. And the same thing for the reverse, if I start from mu 1 and I follow this backward policy, it gets me back to mu 0, or this mu 0 distribution. And these two policies are parameterized by neural networks. So we are actually learning these parameters for these neural networks. And to solve this, this forward-backward theory, we can actually use another more recently developed optimization solution, which uses this forward backward SD, which I was talking about previously, which is like we can iteratively optimize each equation, and each policy has its own loss function. So we do this iteratively and we solve this, and we have this nice solution to both equations. But all this is great. Another problem that we were trying to tackle is modeling the changes in cell population. As I mentioned, cellular processes go through birth, death, and proliferation. So we also want to have that cell population modeling in our system, in our tool. So the cell populations are not conserved, right? The cells as I said, divide and die. So to do this, we use another very recently developed theory, which is this unbalanced version of the diffusion Schrödinger bridge. So very briefly, what that means is that we add another loss term to this iterative optimization, and we're just learning some killing rate for each cell. And based on this killing rate, we can decide whether we want to kill this cell or whether we want it to divide or to proliferate, right? So in this way, our modeling also takes care of the changes in cell population, right? Like if it's a developmental process, the number of cells will eventually evolve. So we want a very small killing rate so that our cells are allowed to divide and we have like this population conservation through the modeling. So with this system, Schrödinger bridges provide a great way to handle the cellular processes. So what we can do with this is we have continuous nature. We can model the continuous nature of the cellular processes. We can handle the stochastic nature. We can incorporate population changes. But there are also problems with this system. There's curse of dimensionality. If you can tell, this is already very complex. Lots of parameters to solve. So this, as the number of cells grow, as the input data set size grows, we have more number of genes. It becomes very difficult to solve. And also Schrödinger bridges are very sensitive to noise. And as we know, gene expression itself is very noisy by nature. So it's just not directly applicable. So to use these advantages and to offset the disadvantages of Schrödinger bridges, we can introduce this variational autoencoder. So with variational autoencoder, we can actually handle this high dimensionality by learning interpretable latent space and also offset the noise. So what we do is, before we use any part of Schrödinger bridge, we just try pre-train our gene expression from observed time points using our VAE, so that we have this nice latent space that can go directly into our Schrödinger bridge system, and we can recover this whole trajectory. So what we do here is during pre-training, we just append this time component with each cell by gene matrix right here. And then we also append a time component to the latent space when this goes into the decoder. So both our encoder and decoder encode some kind of time information, right? So that the latent space also is carrying this temporal information. Adding this time component to the decoder is actually helpful, because when we are reconstructing the unmeasured time points, we can use that time as a cue to the method to kind of approximate those unmeasured time points. And so once we have this pre-training completed, what we do next is just add the Schrödinger system and use the latent variables from each time point and learn this trajectory, and also add another optimization loss term, which is this L joint loss, which jointly optimizes both the VAE and the Schrödinger bridge. So what we're doing is the latents that the Schrödinger bridge predicts for the unmeasured time points, those just go into our VAE, and the VAE uses this unmeasured time information and tries to reconstruct those unmeasured cell by gene matrices. And we also have this VAE reconstruction loss here that also helps to maintain the reconstruction quality of the decoder. So with this method, I will move to the results. So we applied this model to 3 time series data sets with different number of cells, different number of time points, and either these were developmental processes, or there were organ development, embryogenesis, and also to a disease data set. So the first result I want to show you is this performance for unmeasured time points, right? So what we did here was, for these multiple time points, we just removed some intermediate time points, and we tested how well the model could reconstruct those removed time points. And as you can see, ARTEMIS did a good job with most of the time points are close to the highest. And here I just show you one example through a UMAP of what the reconstructed or the predicted time points look like for ARTEMIS and some of the other state-of-art methods developed over the years. On this is an embryogenesis data set with 12 time points. Another result I want to show is what the model is actually learning. So if you recall this, we had these two stochastic differential equations. The one was the forward SDE, which takes us from the starting time point to the terminal time point. And we are learning this forward policy. So what does it look like when we actually visualize this forward policy, right? So this plot here, comparing ARTEMIS to another method, Prishan, we see that we want to see that this policy is some kind of a force field that shows how cells are pushed over time from the starting point towards the end point. And compared to another method, ARTEMIS did a good job with actually capturing this motion of cell development from early to later stages. And what we can also do is, what happens if this forward policies are learned in the latent space, but what happens if we project it to this high dimensional space? And can we learn some kinds of genes, or what genes are driving these processes at different time points, right? So we can actually do that. We can try to project this learned latent forward policy to the gene expression space, and to do that we just multiply this latent policy times the Jacobian of our decoder to get this gene drift score. So this is like an average score across all cells. So for each gene, basically, we have some kind of score based on this learned policy. And when we ranked this in the an example on the zebrafish data set, we found that a lot of the genes highlighted came from these different lineages that were very prominently growing at that time point. And of course, the paper has more examples. Then I also want to show, I mentioned earlier that we are capturing these cell population changes. So the third result on the left, the first plot is actually showing what the model predicted were the cell population changes versus what the ground truth looked like. And in a lot of cases, the model did a good job with recovering these time points, except for this time point 4, which I want to draw your attention to, that was which it missed, because the assumption that we made that the cellular processes are linear and they're not drastically changing. So for these cases where the gene expression or where the population changes drastically, we need some kind of prior knowledge, clearly. And to this plot on the right, I just want to show what the model actually came up with, like how many cells it killed, how many cells were reborn, how many proliferated. And this is very consistent with the plot on the left, where whenever the number of cells declined here, there was an increase in the death rate, or the number of cells that were killed. And to your right, the fourth result is a small experiment to test in silico perturbation effects. So this gene is this TPM1 gene, which is a drift gene identified by ARTEMIS for this EMT process in cancer cells. That also was one of our applications. So in this, what happens if we perturb this TPM1, which is also a prior known hallmark EMT gene? So what we found is that when we underexpress or knocked down this gene, we saw that there was a huge increase in the number of cells. So actually what we did was we knocked down this gene and we allowed the model to predict this trajectory. So when this trajectory was predicted, we saw that the number of cells that it generated were very high for the earlier time points, versus when this gene was overexpressed, we found that a lot of the number of genes were generated towards the later time points. This suggesting that potentially underexpressing this gene may reverse EMT, while overexpressing may promote this EMT progression. And with that, I want to move to highlight some limitations of this work. Of course, the method assumes that there are gradual changes in cell states, and which is in most situations not quite what we see. And of course, there are times when the gene expression changes abruptly, the population abruptly erupts and declines. For that kind of modeling, we need to incorporate some kind of prior knowledge. Then also the assumption that the sequence cell counts are actually the ground truth. Again, as we all know, sequencing experiments are can result in a lot of errors. So again, that assumption is a limitation and needs more prior work to model what is actually happening. And of course, this is a complex framework, so there are a couple of hyperparameters that we need to tune, and again, a time-consuming task. But we found that some of the hyperparameters were consistent across data sets. And to address some of these limitations, future work could incorporate prior knowledge, like including growth rate estimates from known cell cycle or apoptosis genes that can improve the robustness. And we also plan to extend this to multimodal trajectories, where we can integrate other modalities like chromatin accessibility. And so we have like multimodal learning where we can see how two modalities are changing together over time, and also possibly to spatiotemporal data to see how cells change both in space and time. And with that, I would just want to thank my lab, my PI, Dr. Wang, and everyone from my lab at UW-Madison. And also thank you all for attending this talk. And we can take any questions.
Thank you, Sayali, the interesting talks. And it's time for the coffee break, but let's see a couple of minutes for questions. Roland?
So what's the idea for doing a discrete modeling? Like, I didn't understand that part.
Discrete modeling, because the data that we have when we are capturing developmental processes, it's captured at discrete time points, right? So like, for example, you might have some data captured at time 0, then something at time 5, and then time 10, 20. And then what happens in between is kind of missing because when you sequence a cell, it's destroyed, right? So we don't know how it's going to change with time. So we take in the discrete measurements and we try to reconstruct a continuous process. That was the goal of the project.
Okay, but that Schrödinger equation is stochastic, right?
Yes.
So where does the stochasticity come in? I can imagine for an individual cell, the decision is stochastic, right?
Right.
But why not just model it as like a differential equation, like some continuous approximation to the whole thing? Like, it seems like to me this is like overkill to go for the full stochasticity, because if you have only the data as numbers of cells and fractions, relative numbers of different types of cells, as opposed to the individual trajectories of the individual cells, it seems like overkill.
Well, the idea was actually to be as close to the ground truth. How close can we get to the ground truth cellular processes, assuming that they are stochastic by nature, and we can model them through a stochastic process. And Schrödinger Bridge just happened to be a method that could capture this very well. And we have also some important components, interpretable components that we can extract from this modeling, like policies that can highlight for each cell what could have been the driving factor. Like if you see what the model was learning for each cell, which gene was prioritized or not, we could use that as a component, as opposed to some of the other methods like ODEs that could have that potentially don't capture that kind of information.
Have you assessed the sensitivity of your the trajectories that you get to noise and how many sick data? Like try to simulate some noise and see how sensitive they are.
Well, one way we assess that is how close the training points were predicted versus what the ground truth training data looked like, and in that sense, it was quite close to what the training data looked like. Like, of course, we have the noise component in the equation, but the only way we could do that is to see how close the model predicted the training data, the ground truth that it was actually trained on. Like what it does is if you just provide the cells from time 0 and you let it predict the rest of the trajectory, right? So even in that case, some of the points that it was trained on, we just picked those and we compared them to what the ground truth training data looked like. And if that was actually quite close, then that meant that it is good at recovering what it was trained on, apart from what the validation points were. So, yeah.
So thank you, Elaine, for